Welcome you all in the new series of lecture that is the digestion of protein and absorption of amino acids. So here we have a dietary protein which enters into by mouth, but actually in the mouth we don't have any active enzymes that can act on a protein. So there is a no digestion in the mouth, only the mastication process is there. So the protein which enter into your stomach and here it is a denatured. So this denaturation is because of concentrated HCl which is secreted by the stomach. And here there is a pepsin, this pepsin which is also inactive that is activated by HCl. And here there is only denaturation and the protein is partially hydrolyzed by the action of pepsin. Now this denatured protein from stomach, it enters into the small intestine and here the protease enzyme which act on this protein and here we get a small peptides and the amino acids. Now in intestinal lining here these are as we have the small peptides and the amino acids they are absorbed by active transport mechanism and here the amino acid enters into your bloodstream. So this is short summary. Let's start a topic. So about 70 to 100 gram of protein, that is a dietary protein which is obtained from both animal and plant sources and need to be digested daily in an adult. Dietary requirements are mainly required for the tissue repair and the growth. And here, the major source of dietary proteins are milk, cheese, egg, meat, fish, liver, pulses, and nuts. And the casein and albumin. The casein is from milk and albumin is from our egg. So here the daily requirement it should be 1 gram per kilogram of body weight. This is a commonly asked question. What is the daily requirement of protein? So here it should be 1 gram per kilogram of body weight. If your weight is 70 kg, so requirement will be 70 gram per day. So the dietary protein serves the three broad functions. Their constituent amino acid used for the synthesis of body proteins like all plasma proteins, enzymes, hormones, we require amino acids. And the carbon skeleton of amino acid can be oxidized to yield energy. As we know that in the gluconeogenesis, where there is a non-carbohydrate precursors, they are nothing but a carbon skeleton of amino acids. And the carbon and nitrogen atom may be used to synthesize other nitrogenous and non-nitrogenous metabolites. Now here, this is our protein, whenever we cook food or whenever we apply heat, the protein get coagulated. And that coagulated protein which is present in a cooked protein, they are called as cooked protein and they are a denatured protein. So denaturation, as we know, it is responsible for breaking secondary, tertiary, and quaternary structure. And protein, it goes into this pathway, and finally we get a amino acids. And in between, we get a primary derivatives and secondary derivatives. And this thing happened by the action of enzymes. Now, whenever protein which enters into stomach, in stomach, there is a stimulation to release concentrated HCl, that is by a gastrin stimulation, and here there is a secretion of concentrated HCl which is required for the denaturation of protein and activation of pepsinogen to pepsin. Those pepsinogen get converted to pepsin which is also an enzyme and the enzyme which act on a protein. Now whenever this protein which enters into the second part of duodenum where there is a stimulation and there is a release of cholecystokinin and secretin. This cholecystokinin act on a gallbladder and this cholecystokinin and pancreasamine and this secretin which act on the pancreas so that there is a secretion of pancreatic juice which contains various enzymes and that enzyme which act on a protein. Let's see which are the enzymes there. And here this gastrin as we know that which is required for the denaturation protein. Then now in mouth there is no active enzyme, so there is a no digestion. While in stomach we have a pepsin, that is a The inactive form is a pepsinogen, and this pepsinogen get converted into pepsin, and which is secreted by the gastric chief cells. Now this pepsinogen is here. This pepsinogen which get converted to pepsin by the acidic pH, which is provided by concentrated HCl. Now here we have another enzyme, that is our renin. This renin, which is absent in adult, but it is present in a newborns, where they are the newborns who are totally dependent on a breast milk. So if they suck breast milk, it can easily enter into the small intestine so it can be easily bypass the stomach. But here you can see that the renin is there. It prevents this action, and whatever the milk which is taken by that baby, it get clotted. So it is clotted. Clotting action is very important so that it will stay in the stomach for the long period of time, and there is a concentrated HCl and pepsin. They will act on that milk. So the action of renin is a renin which convert casein into paracasein, and this paracasein get converted into calcium paracaseinate. That this process is called as a curdling of milk, and that milk will stay in the stomach for a long period of time. Then the action of pancreatic enzymes. So pancreas secretes trypsin, chymotrypsin, carboxypeptides A and B, and the elastase. And intestine it secretes aminopeptidase, dipeptidase, and tripeptidase. Now here we have two types of enzyme, endopeptidase and exopeptidase. This may be a MUGS question where the difference between two peptidases they may ask, or they may ask what are the examples of endopeptidase and exopeptidase. So endopeptidase they attack on internal peptide bond, that is a trypsin, chymotrypsin, renin, and elastase. So what is the meaning of endo? Endo means that they break internal bond and not terminal bond. So here this enzyme will attack on the internal bond, internal peptide bond, and here we will get a two small peptides. Similarly, exopeptides, that means this exopeptides they act on a periphery of protein, that is aminopeptidase and carboxypeptidase. Aminopeptidase they attack on N-terminal amino acids, while carboxypeptidase they attack on C-terminal amino acid. So the end product is a dipeptidase and aminopeptidase. The end product is a dipeptidase and aminopeptidase. Oh, sorry, these are the amino acids. So exopeptides digest terminal peptide, and here you will get a amino acid because they are acting on a terminal end, so that the amino acid will be liberated very easily. Means liberated means released very easily, not liberated. And here you will get a peptide. So here we are getting small peptides, and here we are getting peptide and amino acids. So this is a this may ask in your images exam. Then here you can see this is a mouth. This is your buccal cavity where the protein is entered. But here there is a no proteolytic activity. In mouth there is a no enzyme, so there is a no proteolytic activity will be there. Then protein it then enters into the stomach, and here the digestion process starts here. So here in protein in stomach we have a chief cells that secretes pepsinogen, and this pepsinogen which is the precursor of pepsin, and here we have a parietal cell that is the concentrated HCl secreting cells, and that is secreted by the action of gastrin. Now we have a pepsinogen, and on which this concentrated HCl will act, and here you will get a pepsin. The pepsinogen get converted to pepsin in presence of Sc concentrated HCl, and pH is 1.5 to 2. So this pH is very important for the activation. Now this pepsin it is a exopeptidase, and this pepsin which act on the peptide bond where the amino acid participate in formation of the peptide bond. So the amino acids are aromatic amino acids and the acidic amino acids. So here this phenylalanine, tyrosine and tryptophan and acidic amino acids are aspartate and the glutamate. Now here you will get a polypeptides, and that polypeptide it will go into the pancreas in a second part of duodenum where there is a cholecystokinin and pancreosamine and secretin. They act on the gallbladder and pancreas to secrete the pancreatic juice, and here there is a pancreatic juice is secreted. And so here in pancreatic juice we have two types of enzyme, endopeptidase and exopeptidase. So here this endopeptides they are in inactive form, that is a trypsinogen, chymotrypsinogen and proelastase. And here we have a exopeptidase, that is a procarboxypeptidase A and B. Now here you can see this trypsinogen which get converted into trypsin by the action of enteropeptidase. Once the trypsin is formed, it ought to activate itself. That is, the trypsinogen get converted into trypsin by the activation by trypsin itself. So this trypsin again it is responsible for activation of or conversion of chymotrypsinogen to chymotrypsin, proelastase to elastase, procarboxypeptidase A and B to carboxypeptidase A and B. Now, now here we have a polypeptide chain. It has an N-terminal end and a C-terminal end, and here it contains peptide bond, that is a CONH bond. So here the amino peptidase, this it act on the terminal amino acid, and they are present on the luminal surface of intestinal epithelial cells and bring out the final digestion of oligopeptides into the free amino acids. So aminopeptidase A and aminopeptidase N, these are the aminopeptidase, they are the non-specific exopeptidase. While the trypsin it is a strong proteolytic enzyme and it is optically active at a pH of 8, and the hydrolysis of the protein into peptones and proteases by cleaving the internal peptide bond. And here involve it involves the amino acid that has the carbonyl group contributed by the alpha COOH group of arginine and lysine. Similarly, the chymotrypsin which hydrolyzes the protein into peptones and proteases by cleaving the peptide bond involving the carbonyl group contributed by alpha COOH group of aromatic amino acids, that is a tryptophan, tyrosine, phenylalanine, methionine, leucine, arginine and histidine. And here we have a elastase. This elastase, this it is also endopeptidase which hydrolyzes the protein elastine by cleaving the internal peptide bond involving the carbonyl group contributed by alpha COH group of alanine, glycine and serine. And here we have a carboxypeptidase A and B. So these carboxypeptidase are the exopeptidase, and there are two types A and B, and these peptidases are metalloenzymes containing zinc and have been included into the zinc peptidase. So these carboxypeptidases which also cleave the terminal peptide bond involving amino group of amino acids. So here there is a amino group of amino acid of valine, leucine, isoleucine, and alanine, bearing a free alpha COH group. So here it acts while carboxypeptidase B, it hydrolyzes the proteins from their carboxyl terminal, that is C terminal, by cleaving the terminal peptide bond involving amino group of end amino acids like arginine and lysine. So here these are the site where this trypsin, chymotrypsin, elastase, carboxypeptidase A and B they act, and this carboxypeptidase A and B they act on the terminal peptide bond, while the aminopeptidase which act on the this N terminal and carboxypeptidase on a C terminal. So this is very important. Try to remember. It may be asked in your MCQs. Now here you can see that at the end you get a oligopeptides and amino acids. Now this amino peptidase, that is a dipeptidase and tripeptidase, they broke that dipeptide and tripeptide, and here you will get a free amino acids. So here we have free amino acids. So let's see the absorption of free amino acid. Now the absorption of amino acids from the distal jejunum and ileum. So now here you can see that this is a intestinal mucosal cell. This is our intestinal mucosal cell in which the amino acid absorb, and here you can see this is a brush border of this intestinal mucosa, and the absorption is depend on the sodium ion, that is the sodium ion dependent carrier mediated transport of L amino acid. Similarly here we have a H plus ion dependent carrier mediated transport of peptides. So here it is a carrier mediated. So here this sodium ion and amino acid which enters inside the cell in this mucosal cell. Similarly, the H plus ion along with this dipeptide or tripeptide, it enters inside the cell. So here this sodium ion and amino acid, they share a common carrier. Similarly, this H plus ion and the dipeptide and tripeptide, they share a common carrier. So here for this amino acid transport, there are near about 7 different type of transport system is available. And here on this brush border, you can see this is a peptidase. These are the dipeptidase or tripeptidase which break that bond between the dipeptide and dipeptide, and we will get a free amino acid, and that amino acid can come from this pathway. Now here this free amino acids, we have a dipeptide or tripeptide on which the peptides will attack, and we will get a separate or free amino acid. So this amino acid, it is then absorbed in a circulation, that is it enters into the portal circulation, and that goes to the liver. Now here you can see that there is the absorption, and now here you can see there is a sodium ion which is elevated in this mucosal cell, so it has to go outside, and here this at the sodium ion and amino acid, they are moved inside the cell against the concentration gradient. This is because here we have a sodium potassium ATPase, and here this 3 plus sodium ion, 3 sodium ion, it enters into the circulation and in exchange with a 2 potassium ion. So here there is a requirement of ATP, and this step, it is a ATP dependent step. So it is a sodium potassium ATPase, it is active here. Because of that, the amino acid and sodium ion are entering inside the cell, and this amino acid, they can be easily enter into the circulation. So here this L amino acid and D amino acid is also enter into circulation. This L amino acid, the absorption is carrier mediated against the concentration gradient, and the absorption of D amino acid is by the simple diffusion. And the absorption mechanism, it is the same as that of D glucose. Thank you.